Okay, in this video we are going to be examining the properties and mechanism of lipid storage in adipose tissue. So fatty acids, recall, are a chemistry problem for the body. What I mean by that is that the fatty acids are amphipathic molecules. Recall what amphipathic means is that both a hydrophobic plus a charged group is present on one molecule. Because of the hydrophobicity, this makes this molecule very insoluble in an aqueous environment. And so therefore we need an organ that is specially designed, specifically able to handle amphipathic molecules. That is adipose tissue. So the design and structure and mechanism of adipose tissue is such that it can shield the hydrophobic nature of fatty acids from the aqueous environment and prevent destruction. This is why fatty acids stored in any other cell type, including liver and muscle, is very, very pathological, because the hydrophobicity of these fats can solubilize plasma membranes, causing cellular lysis and cellular death. So what I want to take a look at in this video is how are lipids stored in adipose tissue. Let's start out by looking at this plasma membrane of an adipocyte. So an adipocyte is the individual cells that make up adipose tissue. Now, for lipid storage, this means that we are not undergoing beta oxidation to any great extent because we are in a fed state. So being in a fed state, we have dietary lipids that we've consumed in our meals, and we also have lipid synthesis, fatty acid synthesis, from excess glucose by the liver. So fatty acids are coming in a fed state from both liver plus dietary sources. So these are the sources of the fatty acids that are going to be stored in the adipose. Now, being in a fed state, that means that we have, of course, insulin signaling, our anabolic signal. The insulin will activate its receptor on the cell surface. And this activation, of course, will perform the same type of reaction as in skeletal muscle. It is going to mobilize GLUT4 to the cell surface. These GLUT4 molecules, these glucose transporters, are located in intracellular vesicles. And so the stimulation of insulin is going to move these vesicles to the cell surface to fuse and now expose GLUT4 on the cell surface. This mechanism is the same as we've seen in skeletal muscle. Now, with high glucose on the outside of our cell, because we are in a fed state, the glucose is going to rush into the cell, just as we've seen with skeletal muscle. The first reaction we'll see is the conversion of glucose to glucose 6-phosphate. This, of course, is being done by the enzyme hexokinase. Now, at this point, the glucose 6-phosphate is going to enter the glycolytic pathway, just as it does with any other cell type. However, although glycolysis will generate ATP and energy for the adipose for its normal cellular functions, the primary function of this uptake of glucose is to generate glyceraldehyde 3-phosphate. Now the purpose of glyceraldehyde 3-phosphate in this reaction, there's two fates for this, of course. This can go on to produce energy for the adipocyte, again, for its normal cellular functions. Or, in this process of the next step of glyceraldehyde 3-phosphate dehydrogenase, this is our dehydrogenase enzyme, recall at this step we are making some NADH. What we can do with this NADH is we can convert glyceraldehyde 3-phosphate into glycerol 3-phosphate. Now recall, this is also a redox reaction, and so we're generating NAD, which can continue to support continued energy production if we need some additional ATP. Now, the glycerol 3-phosphate is going to be serving as the backbone of triglyceride synthesis. So what is triglyceride synthesis? Well, this is going to have a glycerol backbone, and we're going to be adding fatty acids to this structure in order to make a triglyceride, which is the storage component for lipids and adipocytes. Now, where do these fatty acids come from? Recall that our fatty acids are coming from either liver or dietary sources, and they're going to be higher concentration in the outside of the cell because we are in a fed state. So the fatty acids have to enter the cell, and they are going to be doing so, as we've seen before, by the fatty acid transport protein, FATP. This is a facilitated transporter that will allow the passage of fatty acids inside the cell. Now recall that this transporter, being facilitated transport, is able to go in the reverse direction and send fatty acid out, which is counterproductive. So the next step of the reaction we need to do is add CoAsh to this reaction by the fatty acyl synthetase enzyme, and we are going to be making a fatty acyl CoA intermediate. Now recall, this reaction here is going to take quite a bit of energy, to the point we are going to be hydrolyzing ATP all the way down to AMP and a pyrophosphate, and we will further hydrolyze the pyrophosphate to generate even more energy to drive this reaction. So that means that, again, this acyl linkage between the fatty acid and the CoA is a high energy bond. So it's this energy of this bond that is going to be driving the storage reaction for lipids. Now, once we have our glycerol 3-phosphate, we have our fatty acyl CoA. Now we have all the components necessary to generate a triglyceride. And how do we make a triglyceride? Very simple steps. Here is our glycerol 3-phosphate. I'll leave off the hydrogens, let you fill those in your notes. And what we are going to do is a sequential addition using an enzyme called fatty acyl transferase, and we are going to be adding the fatty acid as an ester linkage onto the carbons of glycerol 3-phosphate. Again, the hydrolysis of the CoA off of the fatty acid will generate the negative free energy that we need to drive the reaction. So we are going to take a fatty acyl CoA and add it to here, and to here in 3 successive steps. What we will generate is this molecule. Notice, for example, this is an ester linkage. So this would be the attachment of one fatty acid to the number 1 carbon of glycerol 3-phosphate. Then we are going to do this 2 more times. And what we have in the end is a triglyceride structure. Now notice the triglyceride also called triacylglycerol, also called triglyceride. This structure here is essentially completely hydrophobic. We have long hydrocarbon chains, 3 of them. We do have some polarity in the molecule. There's no charge in this molecule, but we have some polarity with these oxygens. But there's so much hydrophobicity to this molecule that it overwhelms the polarity of this, making this essentially 100% hydrophobic. Now this is going to be our storage form of our fatty acids in adipose. What we are going to do is we are going to add these to a growing lipid droplet, which is located in the cytosol, the cytoplasm of adipocytes. So if you were to look at an adipocyte with light microscopy, what you would see is a very, very large round cell, and you would see a structure pushed to the periphery. That is the nucleus. So the nucleus actually gets squashed to one side of the cell. The central portion of the adipocyte storage is the large amount of triglyceride structure. So this large structure is the triglyceride lipid droplet. Now with this hydrophobicity stored in this lipid droplet, notice how close it gets to the plasma membrane. If we have a hydrophobic substance like this, we could potentially damage this plasma membrane and cause lysis of the adipocyte and kill it. So what the adipocyte is able to do is synthesize a protein called perilipin. Peri mean around, lipid, droplet, around the lipid droplet. So this, let me correct this spelling, it's called perilipin, and it's around the lipid droplet. This protein serves to shield the plasma membrane from the triglyceride structures to prevent lysis of the cell.